Hello, my name is Gershon Levi Ariker, and today I'll be talking about the use of GMO goats to study atrial fibrillation. This is a research project for the biotech course at the University of Connecticut. This is the study that I'll be talking about today. The problem that is being solved here has to do with atrial fibrillation. It's the most common arrhythmia of the heart and is characterized by an irregular, rapid heart rate. As you can see on the left, a typical heartbeat happens at set time intervals and happens quite consistently. While a heart undergoing an episode of atrial fibrillation, these peaks are close together, meaning that it has a higher heart rate, and it happens quite randomly. There's no set time interval between any of these peaks. This also looks at a phenomenon known as atrial fibrosis, where the tissue becomes fibrous in the heart. It is linked with atrial fibrillation and is driven by a gene called transforming growth factor beta 1. However, the mechanisms for the relationship between atrial fibrosis and atrial fibrillation isn't well understood. Why is this? The reason for this was the use of poor animal models. The link of transforming growth factor beta 1 and atrial fibrillation was established in mice, but mice are a bad model for this disease. They have small hearts, meaning it's hard to observe what's going on inside of them, and you can't test treatments on them because of the differences in the cardiovascular system between mice and humans. Larger animals are typically better for this. They have larger hearts, meaning that any phenomenon can be easily observed, and they're also more similar to humans, meaning that any treatment can be transferable between them and humans. Dogs and goats are best because they don't develop natural fibrosis and they're also the proper size. So this study chose to use domestic goats to develop a cardiovascular disease model using the transforming growth factor beta 1 gene. In order to actually create these GMO goat models, they took fibroblast cells from goat fetuses when they were around 27 to 30 days old. They then used electricity to create pores in the cell membrane in a process called electroporation. This allowed them to inject a specific strand of DNA with the transforming growth factor beta 1 gene and drug resistance to a drug called G418 into the cells. As you can see here, this is the gene that codes for the transforming growth factor beta 1, and this is the gene that codes for the drug resistance. Only cells that incorporated the DNA were actually able to resist the drug when it was applied, whereas all the other ones died. This allows them to isolate just the ones that this process was successful in. Now that they had cells where the genome was successfully modified, they are able to use a process called somatic cell nuclear transfer in order to actually convert them into embryos that can be eventually grown into real goats. This was done by taking the nucleus from the cells that the process worked on previously and injecting them into egg cells that had their nuclei removed. These are artificially matured and then implanted in female goats, and eventually they could develop and be born as GMO goats. Once the goats had matured and become adults, the experiment could begin. Goats were given induced episodes of atrial fibrillation and then had multiple analyses done. An electrocardiogram in order to look at the electrical activity of the heart, images of biopsy tissue were observed in order to look for atrial fibrosis, and the gene expression of the goats were looked at just to make sure that the incorporated genes were activating properly. Here we have some results for the experiment. The white bars show wild-type goats, or goats that did not have their gene modified in any sort of way, compared to the gray bars, which are transgenic goats who had their genes modified in the ways we talked about previously. On the y-axis is the p-wave duration, of which a longer p-wave duration is associated with a longer episode of atrial fibrillation. For all months of the goat's maturity, as you can see on the x-axis, the transgenic goats had longer episodes of atrial fibrillation when compared to their wild types, which is what the researchers wanted. Here we have electrical data from the hearts of the goats. Here we have the electrocardiogram for the wild-type goats, and right here is when the episode of induced atrial fibrillation happened. The heart rate increased, and it became sporadic as expected. However, this is even more true for the transgenic goats, as you can see on the bottom here. I can even tell where the peaks are, for example. And also the episode happened for a longer period of time, which is what researchers wanted, because longer episodes of atrial fibrillation means that it can be studied more in depth. Overall, the experiment was a success. It created a better model to study atrial fibrillation, not only because the large goats are better for studying this disease than the small mice that used previously, but also because the goats were more susceptible to atrial fibrillation, which allows for more consistent study. This also created the first model where atrial fibrillation was driven by atrial fibrosis. This way, the relationship can be studied and then that knowledge applied to humans. Also, because the goats are larger and their cardiovascular system is more similar to ours, treatments can be tested on the goats for both fibrosis and atrial fibrillation. Here are the sources that I used for this project. Thank you for listening to my presentation. I hope you enjoyed it.